So let's talk about one of the more common tests that are done in the lab. Um, since we're on the urinary system, talk about urine analysis and then the subsequent culture. Now, hopefully, if you've taken A&P, you've gotten some urine analysis in A&P, but it is one of the more common laboratory tests that we get in the lab. And again, it is used to evaluate the function of the kidneys and other organs, but it's performed in three parts: physical characteristics, chemical analysis, and microscopic examination. Not going too much on this stuff because really this is A&P, but we look at first the clarity of the urine. How clear is it? Normal urine should be transparent, should be a light yellow color. Older samples can be cloudy because either of bacteria or crystals that may be found in the urine. Um, Fresh urine samples can be cloudy due to urinary tract infections. Okay. There may be blood cells, there may be a lot of white cells. So the urine is described as to its clarity. Then we look at the color. We should see basically a pale, light yellow color. Darker means the urine is more concentrated. It could be because of dehydration. You may get red colored due to blood. You may get some unusual orange colors due to antibiotic rifampin. Okay, it will turn your urine just this bright orange, and you will be peeing out the bright orange. Okay, so normally it shouldn't have any offensive odor to it. Any foul odor can be indicative of a bacterial infection. Okay, so what we use is we look at this color chart, and you can see the change, and we do have very specific names. So you won't hear these weird names. It'll either be colorless straw, light yellow, yellow, dark yellow. These are our standard way of listing these. And you can see them in the urine collection bottle running from our lovely little, straw up to an amber color. Okay. Then we look at the clarity, and you can see that this is clear. We can see through it. We can have slightly cloudy, or turbid. We could have red blood cells and things in there. Then we do a dipstick. We do a chemical analysis. And don't go crazy with this. Again, you don't need to know the specific gravity and that kind of stuff, but we do a chemical analysis. We do a specific gravity. It tells us, how much dissolved solids are in that urine, and it gives us a good indication of what's going on. We do this using a spectrophotometer, hygrometer, and then you can see the specific gravity is there. But mainly what we'll want to get into is we do this dipstick to test for those chemicals. And you can see the main ones here for the urinary tract is pH. pH is above 7, you could have kidney stones or UTI. A high pH can indicate a UTI, so that may be a clue that we start looking for things. Normal urine is slightly acidic. Protein can indicate a kidney problem because the kidneys are basically dumping protein. They're not working properly. They should be filtering they should be holding the protein back, and they shouldn't be wasting it. So those are some of the things we look at. We've got a really bad infection, we may see blood. But another thing that comes down here is leukocytes, and it detects the presence of granulocyte esterase, and a positive result does indicate inflammation or infection of the urinary tract. Nitrates, the presence of nitrates in urine is most commonly means a bacterial infection. We'll talk about that in a second. All right, and this is what a dipstick looks like. You just dip it into the urine for for a second or two, make sure all the little pads are wet. You pull it out, and there are times along here, you can't see it, but you read each pad at a specific time, okay? And this is what it looks like. So what we're looking for is, and what usually happens is if we get a positive leukocytes, that purple one, or the pH is really alkaline, some indication we will start looking for particular things underneath that microscopic analysis. Again, we can't do the unknowns, so there's a lot of information that we can get from the urinalysis. And again, normal urine contains nitrates, normally present in foods excreted by the kidneys. Certain bacteria, like E. coli and Klebsiella, convert nitrites to nitrates. So we can pick that up on that dipstick. They use a specific enzyme, nitrate can I talk today? Reductase, can't talk. And these nitrates are not found in a normal urine. So if we get a positive nitrate test, it's pretty indicative that we have a UTI, especially bladder infection. We start looking, okay? We start, we get that dipstick in it and it's turned colors, and we're going to let me go look for bacteria and make sure I see it in the microscopic. A positive leukocyte esterase, as we said, detects an enzyme that is almost exclusively produced by white blood cells, mainly neutrophils, which are your bacterial eaters. And this indicates the presence of white blood cells in the urine, okay? So then we would go and do a microscopic analysis. We would spin the urine down very gently and pour off the liquid, and then whatever is left in the bottom of that tube, we would put on a microscope slide and look at it under the microscope. And this is what we would be looking for, okay? We could be looking for yeast, and you can see the difference. These are red blood cells, but these are yeast. So we would report back to, we would report these yeast. If we see budding on the yeast, right there, we would report budding, okay? The other thing we're going to look, there's another one at budding. If we see bacteria, these are going to be tiny, tiny, tiny little things in a microscopic urine, but they're going to be usually kind of jiggling. So it kind of draws our eye to it if we're looking at it, okay? If we see this, that's indicative we need to do a culture and sensitivity. We need to find out what the organism is and then find out what antibiotic it's sensitive to. So then usually this is done in hematology department of the lab. So then that urine would be sent over, or sometimes we will get both and we just divide it and go ahead and cook. Because a lot of times the doc orders, it's a UA and culture, urine analysis and culture. So we get, the specimen, we'll culture out of it first and then pour it out for the urine analysis because we want the culture first. So urine culture is to check for these bacteria in the samples. And in adults and kids, it doesn't make any difference. We can do, newborns, as long as we get the specimen is good. So, and it is performed when we find these bacteria. So the most common cause of UTI in both inpatients and outpatients is E. coli, overwhelmingly. E. coli is followed by coag-negative staph, again, not Staph aureus, Klebsiella, a Klebsiella species, a Proteus species, an Enterococcus, which is was considered a strep, and then our lovely little Enterobacteriaceae and Enterobacter. So lots of these organisms can cause UTIs. So the first thing that's done when we plate it out, we'll plate it out and we'll do two things. We'll do a colony count streak, and then we'll, as we discussed, plate this urine out on several different plates so we can easily identify the organism. And I wanted to just mention the colony count because this gives us a lot of information. And what we do is we have a special loop. It is 1/1000th of a milliliter. You dip it in and let it fill that loop. And then you streak it down the plate and then start like this and go across and go across and go across all to the end. And then you would inoculate the rest of the plates, put it in the incubator, and the next day you look at it. If the colony count is small, that's usually not indicative of a UTI. Sometimes it can be. I mean, if it's coming from a sterile area, like we have gotten taps of newborns, and then, if you get a tap of the bladder, it's like, no, if anything grows, we're going to report it out. But usually what we want to see, urines can be interesting because you can get a lot of, particularly in the females, you can get a lot of vaginal contamination if they don't clean themselves well when they collect the specimen. But so we're going to look for this. There's a couple things we look for. We look for, is it all one organism or one or two, or is it a whole bunch? If it's a whole bunch, usually the lab will report out, says, it's contaminated. We're just not going to continue. But what you do is you count these, and every little dot is 1,000 because that's what you've diluted it with. So you count these dots, and the colony count will be reported out. If I saw 10 colonies, colony count is 10,000. Usually a colony count of over 100 is indicative of a UTI. And we would then continue and do our sensitivity. So colony count greater than 100,000, considered indicative. Colony counts less. If the patient is symptomatic, maybe we just didn't get a really good sample, or they've been drinking a lot of fluids, so that it's not, because usually with a UTI you start drinking to try and flush it out. So we don't worry about that too much. But what we're going to do is treat it if they're symptomatic. The doc may say, go ahead with the colony count. I don't care what it is, do it. And we would do it. Okay. And that's just, what we do. So I just wanted to take you through this to show you this is what's usually done in the lab behind the scenes with one of the cultures that we may do.